Metabolism and the Evolution of Social Behavior. This study looks at how metabolism affects the evolution of social behavior, which occurs on a timescale of seconds, minutes, or hours, and how social behavior affects the genome of a population through metabolism, which occurs over generations and is therefore harder to study. Bacteria make an excellent model because they reproduce quickly. The bacteria used here is Pseudomonas aeruginosa. Social behavior is an interaction among members of the same species that influences their survival and reproduction. There are 4 basic types of social interaction: mutual benefit, selfishness, altruism, and spite. The social behavior studied here is swarming, which is the coordinated movement of bacteria across a surface. Bacteria will signal each other to begin secreting rhamnolipid biosurfactants, which spread out and create a lubricating film. This allows billions of cells to swarm and access new nutrients. To avoid wasteful secretion and exploitation, Pseudomonas aeruginosa uses a system of regulation that ensures the genes for rhamnolipid secretion are only expressed when the population is large enough and individual cells have access to excess carbon source. This control is important because secreting rhamnolipids is a huge investment for each cell. Rhamnolipids are carbon-rich, and bacteria can dedicate more than 20% of their biomass to rhamnolipid synthesis. However, cells that cannot produce any of their own rhamnolipids can still swarm by taking advantage of the rhamnolipids produced by other cells. Wild-type Pseudomonas aeruginosa was turned into the highly altruistic mutant delta CBRA by knocking out a gene. When green-labeled delta CBRA was put on a swarming plate with red-labeled delta RHLA, a strain incapable of swarming, the number and frequency of colonies per strain before and after swarming show that delta CBRA is highly altruistic. 6 populations of delta CBRA were put through 7 days of swarming, which can be divided into 2 phases. One, the parallel phase, marked by a quick recovery of swarming in all populations. 2, the divergent phase, marked by some populations swarming more and some populations swarming less. Sequencing of isolates showed that all colonies had at least 1 mutation in either the CRC or HFQ genes. Wild-type Pseudomonas aeruginosa was turned into the altruistic mutant delta CBRA. After further experimentation, delta CBRA produced new mutants, delta CBRA-CRC and delta CBRA-HFQ. Delta CBRA-CRC was more common, and more similar to the wild type's metabolome, but less evolutionarily stable due to its altruistic behavior. Delta CBRA-HFQ was less common, and less similar to the wild type's metabolome, but recovered its social behavior and was more evolutionarily stable. This research implies that social behavior can have an impact on metabolism. For example, some people become more aggressive when hungry. The ratio of those who behave aggressively to non-aggressively will change over time based on which behavior presents the greatest advantage.